everyone. I am Professor Charu Dogra from Ramjas College, University of Delhi, and I would be continuing with the newly started series on Microbial World. We were discussing about the overview of microbiology, in which we had talked about the different disciplines under microbiology, and we also started with talking about a little bit history about how this microbiology came into being and the contributions of certain scientists. So, to continue with it today, we will be discussing some more about the history of microbiology and contributions of some renowned scientists. Two of the very important scientists we will discuss today. We will also understand the biological classification for understanding the microbial diversity. The references used for this session for educational purposes are the Pelkzer Microbiology and the Microbiology and Introduction by Tortora and et al. The first golden age of microbiology, as the chronology is given on your screen, there were many important scientists who worked on it, and we discussed this in detail in our previous sessions, where we talked about the contributions of Antony van Leeuwenhoek, where it all started from, the animalcules, and then we also talked about the contribution of Louis Pasteur. They basically established what is called as the germ theory of disease. So, that is where all this microbiology started from, that they obtained that these invisible, these small tiny creatures were causing the diseases, various kind of diseases. And many of these scientists proved this, right, starting from that there were certain rotten meat and how it can happen. They first thought that it is because of the elemental things, like because there is air which is present and therefore it is happening. But initially, they thought these things, but when they experimented, this zeroed down on the germ theory of the diseases. So, many contributions, many scientists are given here, how the cell theory came into being, and gradually it moved on from there. We talked about the Koch's postulates, but I will quickly go through it once again, that Robert Koch, he basically had a diseased mice, and then he isolated certain cells and the microbes which were causing the infection. So, he cultured them on the plates and also identified them under the microscopes. He then basically injected this microbe into a healthy individual, and gradually the disease was manifested in that healthy individual as well. Now, to ascertain that this disease is because of the same microbes, he again plated the samples from the healthy turned diseased mice, and he identified the microbes which were present in there. When he compared the microbes which were initially present in the diseased organism and which were introduced into the healthy organism which made it diseased, then he found that both the organisms were similar, the same microbial communities were there. So, Koch basically postulated that the same pathogen must be present in every case of the disease. So, he basically said that whatever a disease is manifested, then it is caused by a microbe which is harmful to a body is called as a pathogen. So, he basically postulated that because the same pathogen could be identified from the diseased organism and it could be identified from the healthy turned diseased animal, therefore, the same pathogen must be present in every case of the disease. The pathogen must be isolated from the disease host and grown in pure culture. So, in the experimentation as he did, he postulated that the pathogen must be isolated from the disease host and then grown in pure culture form. The third postulate was that the pathogen from the pure culture must cause the disease when it is inoculated into a healthy, susceptible laboratory animal. So, what he postulated, why did he give these postulates? He postulated this because of identifying various microorganisms for causing various kinds of diseases. So, at that time, there were no association, there was no germ theory of diseases. So, many diseases were there which were manifesting. And then how to ascertain that a particular pathogen is causing that disease, he postulated these steps. So, the third step was that third postulate was that the pathogen from the pure culture must cause the disease when it is inoculated into a healthy susceptible lab animal, and therefore, one can actually conclude that that particular disease is caused by the pathogen which has been injected and isolated from the diseased one. So, the fourth postulate was that the pathogen must be isolated from the inoculated animal and must be shown to be the original organism. So, if all these postulates are met, then one can ascertain the causative microorganisms for various diseases. Now, there were many exceptions to Koch's postulates. For example, the Treponema pallidum, it is known to cause syphilis, but virulent strains have never been cultured on artificial media. So, the basic limitation of Koch's postulate, why these exceptions are there, is that not all pathogens are culturable. There was a very prerequisite for the microorganisms to be cultured. Cultured means to be grown on the artificial media. We have discussed in detail about this in our biotechnology series. You can always refer to that how the media is prepared, how the microorganisms are cultured, etc. So, such organisms are exceptions because the virulent strains have never been cultured on the artificial media. The causative agent of leprosy, which is Mycobacterium leprae, has been also never been grown on the artificial media. Rickettsial and viral pathogens, they cannot be cultured on artificial media because they multiply only within the cell. So, the viral pathogens, because they need a host to multiply. So, again, without host individually, they cannot be cultured on the cell, on the plates. Use of tissue samples from the diseased, advanced microscopy, immunological techniques. So, all these poses certain kind of limitations and there were exceptions to it. Because it was difficult to obtain the tissue samples from disease, there were not advanced microscopy techniques which were there or immunological techniques which were there. So, all these things could be used to identify the microorganisms apart from doing the cultural techniques. So, these limitations could be overcome by many of the advanced techniques which are there. Then some infectious diseases are not as clear cut in their signs and symptoms. This is again a very important point to understand, that what happens is there is not a very clear cut cause and effect connection. It's very difficult sometimes that the infectious diseases, the signs and symptoms it is manifesting, there are some very common symptoms which are there. So, one cannot actually ascertain that the infectious disease, what kind of infectious disease is there, or how to ascertain the organism associated with it. So, for example, nephritis, which is the inflammation of the kidneys, pneumonia, meningitis and peritonitis, these diseases, you cannot just ascertain only one sign or a particular sign or symptom with these particular diseases. So, like inflammation of the kidneys, many different diseases can cause this. So, just for the nephritis, what is that diseased? How would you ascertain that the disease is there or this particular disease is there? So, that is also one of the exceptions for the application of the Koch's postulates. Then some pathogens can cause several diseased conditions. For example, Mycobacterium tuberculosis, implicated in diseases of the lungs, the skin, the bones and other internal organs. Or Streptococcus pyogenes, it causes sore throat, scarlet fever, skin infections and osteomyelitis, which is inflammation of the bone, among other diseases. So, again ascertaining one disease to only caused by one microorganism or one microorganism causing only one disease using the Koch's postulates, there are many exceptions, many challenges and many limitations. However, when we are studying introduction to microbiology, it is very, very important to know the contribution of Koch's postulates, because in very simple and straightforward diseases, they are very, very useful and important for ascertaining the microorganism, the disease causing microorganism or a pathogen causing a particular disease. Ethical considerations may also impose an exception to Koch's postulates, because when we are saying that you inject the organism from a disease to a healthy organism. So, injecting any diseased thing to a healthy individual causes the lot of ethical considerations, because to ascertain that we are basically killing a healthy organism. So, for studies it's okay, but again there are lot of ethical considerations which are imposed. For example, in human experimentations with untreatable diseases, they are unacceptable today. You can't do inject the particular microorganisms which are yet untreatable. To find the treatment for them, you cannot conduct experiments on human. It's unethical because, again, it's a cause of concern that you cannot inject any pathogen which is untreated right now to conduct on humans. So such kind of things, causes exceptions to the court's postulates, but otherwise, if it's a very straightforward disease causing agent and there is very clear cut symptoms and signs, then following the court's postulates, one can easily ascertain the microorganism or the pathogen causing a particular disease. So now we discuss about two pioneering scientists whose discoveries revolutionized the medicine and saved millions of lives worldwide. Now would you see that how this microbiology, we are also discussing the history of microbiology. So how slowly and slowly, parallelly, when the many microbiological advanced techniques were also discovered, then how all these things came into being. More and more microorganisms could be discovered, and then there were certain important applications of these microbes, as we will see in the vaccination and antibiotic generation. The work which was done by these two pioneering scientists, that how it revolutionized even the medicinal areas and saved millions of lives worldwide. Because what happened is that during those times there were many pandemics or many diseases, which there used to be outbreaks of such diseases, and nobody knew the cause of that. So because as the microbes are they are invisible to our eye, and they didn't know, they were just discovering these microorganisms or associating these microorganisms with various kinds of diseases. So initially what they used to do was, before finding out the mechanism how the disease is caused or what microorganism is cause a particular disease, they used to find the treatment, because the main idea was to save the individuals who were suffering from a particular disease, or if it is an outbreak, the masses of people, the population. So the idea was that. So despite knowing the underlying mechanisms, how the disease is caused, they used to also concentrate their research on how to find the treatment for those particular diseases. So let's discuss about the contributions of these two important scientists. First one is the Edward Jenner. He is called as the father of immunology. You must have heard of his name. He's a very important person, and during the COVID times, even the people who didn't know about Edward Jenner would have come to know, because there was a lot of discussions and debates, and everybody, the layman, even not from the scientific background, came to know about the vaccines, although they already know about it, because there are certain vaccines which are given to individuals during their growing ages. The kids are given different vaccines, but this name was very, very prominent when the discovery of new vaccines was concerned, and during the COVID pandemic, even the non-scientific people came into knowing all these scientific things. I have already discussed that in my previous session as well. So Edward Jenner is the father of immunology. He was born on May 17, 1749 in Berkeley, England. His revolutionary achievement was he pioneered the concept of vaccines and created the world's first smallpox vaccine. Now, before I discuss about his contribution, I would like to put across a very important point, which I always emphasize, that science starts from curiosity and observation. So it is very important, anybody can be a scientist. The only thing is that you need to have a keen eye for observation, and you have to have that kind of curiosity which is there. That is, in very sophisticated terms, that is one of the component of what we call as scientific temper or scientific temperament. I mean, these are two distinct things, but just to, tell in a very broad manner what is called as the scientific temper. So, that curiosity, that is intrinsic in our, attitudes. The only thing is, if we nurture this kind of observation and curiosity, miracles can be born. You will just observe that these people, these scientists, and they had a very keen observation points. And then they have a lot of curiosities, and they ask a lot of questions. So all the aspiring students who really want to get through and do good in research, please sharpen your observation. Please, be curious, ask lot of questions. There is loads of information which is present even on the web, or your teachers can tell you. But this history, when you come to know about the science, you appreciate the fact that how small and very simple, very straightforward things revolutionized the world of science, and how it helped for the human welfare. So this is one example, and again I am emphasizing on the fact that obviously the people who are listening to these sessions would be interested in science and probably pursuing science. So always remember these points, and these are therefore very important. So how does he pioneer the concept of vaccines? So there was smallpox outbreak, and it was very devastating. There was no cure for smallpox. Complete areas, districts were, the people were getting eradicated. They were dying because of this smallpox virus. And what happened was that this was a very devastating pandemic. The smallpox scourge, if we discuss, it terrorized the humanities for centuries, causing severe disfigurement, blindness and death. The disease spread rapidly through populations with no known cure. So it was a very infectious disease, and it just spread across the people, and people just didn't know how to treat them. There was all kind of disfigurement, like if you see in this slide, there is a normal bone structure which is shown, and then Paget's disease, the disease structure. So a lot of disfigurement was there, there was blindness was there, the people were dying because of this smallpox, and whenever there was an outbreak, for example in the 18th century, there was smallpox claimed approximately 400,000 lives annually in Europe, in Latin America, and those areas. And they were, entire communities were just decimated, and survivors have, permanent scars. So you could imagine that how needful it was to find its cure, and how scientists or the people were just putting their brains into how to get that. And it was just a simple observation that led to the discovery of the curing for this particular virus, and it has now been, removed. There is no, it has been eradicated basically, the smallpox disease. So that is the kind of, impact it had when the outbreak was there. So what did Jenner do? So Jenner's breakthrough came from the cowpox diseases. So what he observed was, so observation is the first thing. He was told by this milkmaids also. So but obviously he listened and he observed. So he noticed that milkmaids who contracted cowpox, a milder related disease, never developed smallpox. So that was his observation. People are dying all along. But the people who had contracted cowpox, which was a milder related disease, he observed that they never developed smallpox. So then he created a hypothesis. It is very important. That is why I put it into steps here, that there is an observation, then there will be a hypothesis, then there will be a test, there will be a research to, experimentation to prove your hypothesis, and then there will be a reiteration. This is the research process. He did it, centuries ago, and you do it now. It follows the same process. The only thing that differs is the problems, the complexity of the problems, the techniques, the tools, they become much more advanced, but the idea remain the same. So what he did was, the first key point was the observation. He observed, he listened to the milkmaids, and he saw that there was a very milder variant, or not the variant, a milder virus, a related disease was there, which is the cowpox, and who contracted cowpox never developed this smallpox. So as I said, the second thing was, what he did was the experimentation. Of course, before that he did was the hypothesis laying down, that he must have laid down this hypothesis that something from this, cowpox disease is preventing the smallpox proliferation in the human beings. So what he did was, in 1796, he inoculated an 8-year-old boy, James Phipps, with the material from a cowpox lesion. So there was a cowpox lesion and from that blister he took some material, and obviously to the volunteer, this 8-year-old boy was a volunteer, so he just injected that cowpox lesion material into him. So he inoculated this into him. Obviously he showed the symptoms of the cowpox disease. He had this mild infection, he had fever, and but he soon recovered, because cowpox is a relatively milder infection which was there. So he recovered. So after 2 months, what he did was, that is what he tested, his hypothesis test, that what he did was that after 2 months, he exposed the boy to smallpox, and he remained completely immune to the deadly disease. So this was the breakthrough, What he did was that he took some smallpox and again inoculated into a cowpox diseased, which he had 2 months ago, boy, and he injected this smallpox virus into him. And he observed that he remained completely immune to the disease. So what he was observing in the communities around, that those who had contracted cowpox were not suffering with smallpox, now he tested in his lab. And obviously just not the one, but this basically proved that this cowpox disease is preventing the occurrence of the smallpox disease in the individual. So the result was, this groundbreaking experiment proved immunity could be artificially induced, creating what is called as the world's first vaccine. So you already know about the vaccine, how it works. There are many types of vaccine, we are talking about that some other strain is helping or creating, because it was a related strain, The body could not distinguish between the cowpox virus or the smallpox virus. Now we know how the immunity works. So what happened was, when the cowpox virus is or a person is contracting cowpox, he is generating antibodies which is against the cowpox virus. But because the smallpox virus is so similar to cowpox virus, when the smallpox comes, our antibodies which were generated against the cowpox works against the smallpox as well. And therefore it does not allow it to manifest the disease. So that is basically one type of reaction that occurs, and one kind of vaccine where another strain is helping. Sometimes we use the avirulent strain of the diseased in organism also. Sometimes we use only a particular unit against which the antibodies can be raised, which is important for when the organism to give immunity when the organism contracts. So, different types of vaccines which are there. In fact, you must have also come to know about the 2 different types of vaccines when the COVID was there, the Covishield and the Covivax. So, that is basically the different types of vaccines which were there. So, this groundbreaking experiment provided the immunity. Obviously, there are many ethical considerations and many concerns also by injecting the avirulent strain, or what is the probability that still it will cause the disease or the dead organism or some other organism. Then obviously you were already inducing a healthy individual by the cowpox strain as well. So you know that it causes a milder infection, but you might not have known that it may be causing some other deformities as well. So there is always a debate that goes on for the vaccines, but of course it saved a lot of individuals and removed the smallpox disease globally. So these kind of things are very, very important. So birth of vaccination comes from Jenner's experimentations. He coined the term vaccination from the Latin word vacca, which basically means cow, because it was based on his work with the cowpox. And then Louis Pasteur in 1880s used vaccine to broaden and cover other lab produced immunizations such as against the rabies and anthrax. So, as I said, different types of vaccines using different mechanisms were then raised to create the vaccine immunization against the lot of harmful diseases such as rabies and anthrax. So, this is the birth of vaccination and the contribution of Edward Jenner. Jenner's work established the scientific basis for modern immunology and preventive medicine, and his work led directly to the eventual global eradication of smallpox in 1980, the only human disease ever completely eliminated. So again, you can understand the importance of such kind of discoveries and experimentations, and also appreciate the kind of research process that goes into such discoveries. It is very, very important. The second key contributor that we discussed today is Alexander Fleming. He is the accidental antibiotic pioneer. So again, accidental, it's a very important word, because accidents, or the serendipity as we say, is because still he had that keen observation. If he would not have had that keen observation, he would have just, it was just a fleeting thought that would have come, or a fleeting glance that he would have given, and this could not have been discovered at that time. I mean, later maybe, but at that time. So again, observation is the key, curiosity is the key, asking questions is the key, then thorough knowledge is the key, that have your base strong, and then go for experimentation and applying things, and then troubleshooting is very important in research process as well. So I am just guiding you along with these, that how these scientists discovered such phenomenal things, and how basically one can do that and follow the research process. So Alexander Fleming, he was born in 1881 in Scotland. He was a Scottish physician and microbiologist specializing in bacteriology. He discovered what is called as penicillin. You must be knowing that, but I will just quickly go through how this was discovered. Fleming returned from vacation to find a mold which became later known as Penicillium chrysogenum had contaminated one of its bacterial culture plates. So as I said, the key is the observation. He had gone for vacation, some plates were lying in the refrigerator. So when he came back, he just looked at the plates, some were outside as well, and he then observed that certain mold was growing onto it. And his observation was that, as you can see, that around the mold, the growth of the organism was inhibited. So he basically concluded that this particular mold is releasing certain inhibitory substance which is inhibiting the growth of the microbe which is there. So it is basically an antibiotic. Biotic means living organism, anti means something which can kill that living organism. I mean, either kill or arrest its growth, that we will come to talk about later, but antibiotic means which is against the living life. So around this mold, you can see there is a halo, there is a clear zone which is there, and what that clear zone signifies is that it was inhibiting the growth of the particular bacteria which was there. So this gave him an idea that there are antibiotics which are produced by certain organisms. He isolated this and termed it as penicillin. This was the substance which was basically the world's first true antibiotic. So we continue with more of this in our next session. Thank you.